Hi, so this is Abhijit Sahu, and today we'll be discussing about biomolecules. In particular, we'll be talking about protein as a biomolecule, its structure, function, and the analytical techniques to visualize and study about proteins. So, to start off, let's have an overview of biomolecules. Biomolecules are organic molecules which include carbohydrates, proteins, nucleic acids, and lipids. Just to have an overview, carbohydrates, also known as polysaccharides, which are consistent of monomers called monosaccharides. Nucleic acids are another type of biomolecule that make up DNA and RNA. Uh, Nucleotides is a constituent of nucleic acids. Nucleotides in itself is divided into nucleosides, which are pentose sugar nitrogenous base without the phosphate. With the phosphate group, the nucleic acid is termed to be as a nucleotide. Uh, the nitrogenous bases that include in a nucleotide are purines, pyrimidines. Purines, like we see, include adenine and guanine. Pyrimidines include cytosine, thymine, and uracil. Other than that, lipids, they consist of glycerol and fatty acids. So, carbohydrates. Carbohydrates are organic compounds composed of CH and O, carbon, hydrogen, and oxygen, typically in a ratio of 1 is to 2 is to 1. So glucose, for example, has C6H12O6. Classification of carbohydrates are broadly classified based on the number of sugar units they contain: monosaccharide, disaccharide, and polysaccharides. Glucose here, as you can see, is a monosaccharide. Sucrose is a disaccharide. The sucrose is formed with uh, two glucose molecules attached with a glycosidic linkage. Polysaccharide, Uh, an example of them are amylose starch, which is a continuous chain of monosaccharides. The primary function of carbohydrates is to serve as the primary source of energy in the body, thus called the energy currency of the cell. Carbohydrates also act as storage molecules in the form of glycogen and starch in case of plants. Carbohydrates are also found in the structure of cells. Nucleic acid are macromolecules that store and transmit genetic information within cells. They are polymers made up of repeating monomer units called nucleotides. There are two primary types of nucleotides, DNA and RNA, out of which DNA stores genetic information and RNA utilizes the genetic information for transmission, protein synthesis, and regulation of gene expression. Lipids. Lipids are a diverse group of organic molecules that are generally insoluble in water but soluble in organic solvents. Some constituents of lipids are fats, oils, phospholipids, and steroids. Primary function. The primary function of lipids is protection, insulation, and slow release of energy. They also act as structural components of cell membranes. As we can see here, a lipid, an example of a lipid here is glycerol and three fatty acid chains, which form a bond together, creating triacyl glycerol. To revise this again, here is a function: fast release energy, structure. These are the functions of carbohydrates. Lipids are the function of protection, insulation, slow release of energy. Nucleic acids encode information in the form of nucleotides and proteins. Proteins have a function of speeding up reactions, enzyme catalysis, um, forming different structures and signaling for the release of different sorts of enzymes. The sources of each of these biomolecules is mentioned. Structural and various forms of proteins. So proteins are generally characterized by four levels of structure. The primary structure is a linear sequence of amino acids in a polypeptide chain. Even a single change in the primary sequence can drastically alter the protein's high-order structures and its function. Example in sickle cell anemia. In sickle cell anemia, a change of even a single sequence of a peptide in the peptide chain can cause the manifestation of sickle cell anemia. More about it is mentioned in the PDF. Structure, the secondary structure, it is a localized regular folding patterns of the polypeptide chain. It is stabilized by hydrogen bonds between the atoms of the polypeptide backbone. Tertiary structure. The overall three-dimensional shape of a single polypeptide chain resulting from the further folding and coiling of the secondary structures. It is stabilized by various interactions between the R side chain and the amino acid, including hydrophobic interactions, ionic bonds, hydrogen bonds, disulfide bridges, van der Waals forces, etc. The quaternary structure is an arrangement of multiple different polypeptide chains which exist in their tertiary structures. This forms a larger, more complex protein. Not all proteins require to exist in a quaternary state. It only applies to proteins made of two or more polypeptide subunits. These subunits can be identical or different, and the formation of these polypeptides also helps increase the stability of the protein further. So let's study about the details of a primary structure of a protein. So each amino acid is connected to the next by a peptide bond, as you see here, which is a covalent amide linkage formed between the carboxyl group of one amino acid and the amino group of an adjacent amino acid. When you see this phenylalanine being magnified, you can see the amino group to the left and the carboxyl group to the right. The highlighted part is the R group, and this is the variable region for every 20 amino acids. Um, the linkage of the peptide bond, as mentioned, it starts at the N terminus, and at the N terminus, this carboxyl group forms a bond with the N terminus or the amino group of the next amino acid in sequence. Uh, with the release of water molecule, when these bonds are formed, H2O is released, and thus the synthesis of these peptide chains is also called as dehydration synthesis. As you can see here, the N terminus and the C terminus. The N terminus stands for the amino group that is the amino group, right? And the C terminus stands for the carboxyl group. The N terminus marks as the initiation point as the start of the polypeptide chain, whereas the C terminus marks the end of the polypeptide chain synthesis. Uh, the N terminus may contain signaling peptides that mark the protein to deliver them to special cellular compartments such as the mitochondria or the Golgi apparatus. Um, C terminus plays a role in the proper folding of stability of the tertiary structure. The N terminus carries a positive charge, as we can see here. The protonated amino acid group here carries a positive charge, whereas the carboxyl group carries a negative charge over here near the single bonded oxygen. It is the deprotonated form as it carries a negative charge. To move on to the secondary structure, the formation and stability of the secondary structure is almost entirely dependent on the hydrogen bond. A hydrogen bond here can be seen between the amino acid and the carboxyl group. The hydrogen and oxygen react to form a hydrogen bond, and the groups of these peptide backbone, alpha helix and beta pleated sheets regularity is due to the inherent geometry of the peptide bond and the optimal formation of hydrogen bonds. So these are the two most common types of bonds that are formed. From these, the alpha helix bond is stabilized by intramolecular hydrogen bonds between the carbonyl oxygen of one amino acid and the amide hydrogen of another amino acid. 4 residues down the I plus 4 rule. So the I plus 4 rule is what helps in forming these even helixes. Right-handed coil. Most helices found in the protein are right-handed. residues per turn, approximately 3.6 amino acid residues per turn. So, so carbonyl, beyond this C double bond O, there is another O towards the back of this helix. And after this amino group, there is another oxygen present behind the helix that we see towards the front. Each turn consists about 3.6 amino acids per residue. Each turn advances the helix by about 5.4 angstrom. Now the beta plated sheets. The parallel beta plated sheets adjacent run in the same direction, N to C direction. The image you see here is not the parallel beta sheet but the anti-parallel beta sheet. They run adjacent in the opposite N to C direction, more common. As we can see here, it is more common and more stable due to the optimal hydrogen bond geometry. When it comes to the parallel beta sheet, the bonds that are formed are not linear, but at an angle. This reduces overall stability and the configuration of the molecule. The pleats arise from the tetrahedral geometry around the alpha carbon. Side chain orientation R groups project alternatively above and below the plane of the sheet. Uh, the tertiary structure forms by the further folding and coiling of secondary structures, alpha helices, beta sheets, turns, and loops. As you can see here, the helices and the beta plated sheets. The stability of the structure mainly depends on the secondary bonds, including hydrogen bond, the hydrophobic bond, ionic bond, van der Waals process, etc. R group interactions occur between residues of far apart amino acids in the same protein, leading to folding and coiling. Uh, that is the tertiary structure of the protein, and besides that, there are two other types of proteins that are classified into globular proteins and fibrous proteins. Globular, globular proteins are the most common type of proteins. They are compact, spherical, and nearly spherical, and highly soluble in aqueous environments. Their compact shape usually means their hydrophobic water-bearing amino acid side chains are tucked away in the interior, while hydrophilic chains are on the surface interacting with water. Globular proteins are usually also occurring in the enzymatic form. Fibrous proteins. These proteins are typically elongated, strand-like, and sheet-like, and generally insoluble in water. The structure often consists of repeating units of secondary structures like multiple alpha helices or beta sheets assembled into long fibers or sheets, giving them high tensile strength and elasticity. These types of proteins are often found in muscle, hair, etc. So, more about fibrous proteins and globular proteins. Let's start off with globular proteins. It is a compact, roughly spherical shape. It is soluble in water typically. It is involved in dynamic and active functions, example catalysis, as it acts as an enzyme, transport. The structure complex. The structure is complex and intricate foldings of various secondary structures to a compact 3D shape, often with a hydrophobic core or a hydrophilic surface. Examples are enzymes like amylases, which break down amylase, hemoglobin, which transport oxygen, and autobody and antibodies, which act as immune defense. Fibrous proteins. Elongated, tough, and often rope-like, sheet-like, generally insoluble in water, provide structural support and protection, composed of long repetitive sequences of secondary structures like continuous alpha helices and beta sheets, collagen in skin, tendons, keratin in hair, nails, etc. So as we see here in globular protein, there's a red spot over here, which is the active site. We'll get more into the active site in the further slides. After the tertiary structure of the protein, there is a quaternary structure of the protein. This structure only applies to proteins made of two or more individual polypeptide chains, which can be identical or different. Each chain is called a subunit. Subunits interact primarily through non-covalent forces such as hydrophobic interactions, hydrogen bonds, and ionic bonds. These bonds are weaker but several of these bonds together lead to the formation of dimers, trimers, tetramers, and pentamers. An example of a tetramer would be hemoglobin. Sometimes covalent bonds, disulfide bridges can also link subunits. Examples of quaternary structure: uh, tetramer for cooperative oxygen transport, which is hemoglobin. Antibodies, four polypeptide chains, again a tetramer. RNA polymerase, large multi-subunit enzyme for gene transcription. ATP synthetase, more complex molecular machine for ATP synthesis. Viral capsid, protein shells of viruses formed by self-assembly of subunits. Enhanced functionality. What does quaternary structure provide to the protein that it needs to exist in such a subunit form? Uh, Cooperative binding. Binding of a molecule to subunit affects binding affinity of others. Example, hemoglobin's oxygen binding. Allosteric regulation. Binding at one site of a subunit affects activity at a distant site of another subunit. Increased stability. Multiple subunits can lead to a more stable overall structure. Formation of active sites. Sometimes active sites is formed at the interface of multiple subunits. Efficient metabolism allows for large enzymatic complexes or streamlined functions. Role of proteins in sustenance of life. Enzymatic catalysis. So as you can see here, the substrate bonds with the enzyme's active site, and the active site supplies some pressure under which the bond it uh, undergoes stress and it breaks down and leads to the formation of an enzyme substrate complex. After the complex is formed, the enzyme releases the products and the substrate is then converted into a product. Similarly, the substrate also can bind to the enzyme's active site, and then another enzyme substrate complex can be formed. The enzyme substrate complex then leads to the merging of the substrates, leading to the production of a final product. Metabolite transporters. So, on the cellular surface of microbes, metabolite transporters exist in the form of integral proteins, peripheral proteins, and surface proteins. These proteins allow the passage of selected molecules. They allow the diffusion of the molecules from within the cell to the outside. They also allow uh, the reverse to happen. The protein channels, also known as transport proteins, allow the passage of certain ions and whenever the need be, certain toxins. Regulation of metabolic pathways. So I'll add more about this in the PDF. Storage of specific nutrients. And this would be the example of ferritin, which is a molecule that stores iron and it releases it whenever the need be for the cell. Some other uses of proteins would be in immune response. The antibodies formed are completely made up of proteins with a few other biomolecules. Uh, signaling molecules. Signaling molecules allow the binding of the signaling molecules bind rather to the open channels of the pores on the cell. This allows the cell pores to open and close depending on the requirement. Now, onto the techniques to isolate and analyze proteins. Gel electrophoresis or SDS page, which is a further extension of gel electrophoresis. To separate proteins based on their size or charge and to estimate their molecular weight is the purpose. Uh, Proteins are loaded onto a gel like polyacrylamide for page and subjected to an electrical field. In SDS page, proteins are denatured by the SDS and coated with a negative charge, so they separate solely by size with smaller portions. migrating faster through the gel, facing less resistance during the run. Applications. The gel electrophoresis can be applied while assessing protein purity, estimating molecular weight, identifying proteins, etc. Western blotting. Western blotting can be used to detect specific proteins in a sample after electrophoresis. Western blotting test was often used for and is still often used to test for typhoid. So, in Western blotting, proteins separated by SDS page are transferred onto a membrane. The membrane is then probed with specific antibodies that bind to the specific target proteins. This leads to a formation of complex, and in order to detect these complexes, rather, they are usually dyed with these tracking dyes, which can be fluorescent or chem chemiluminescent. Uh, the application of these could be to identify protein, quantifying protein, and verifying the expression of protein, by which we can identify if the disease is present or not. Ultraviolet visible spectroscopy uh, measures how much UV or visible light a sample absorbs to identify or quantify substance based on the unique light absorption patterns. A light beam passes through the sample. The instrument then compares the initial light intensity to the intensity after passing through the sample, calculating absorbance, which directly relates to concentration. Commonly used to estimate protein concentration, example at 280 nanometers due to aromatic amino acids, and to assess the purity of biological samples by checking absorbance ratios. These absorbance ratios can later be plotted in a graph, and the highest absorbance, or the lambda max, can be obtained through noting various different samples. Affinity chromatography. So, affinity chromatography is not the only type of chromatography that can help with identification of proteins. Uh, there also exists ion exchange chromatography, which depends on the protein size, and it solely depends on the protein size rather. But let's study about affinity chromatography. In affinity chromatography, a specific ligand, example an antibody or a substrate analog or iron metal, is covalently attached to the stationary phase. The stationary phase is towards the ends rather of the tube. Once proteins are specifically bound in this ligand, they will be retained on the column. Unbound proteins are washed away. The target proteins are then eluded by adding soluble forms of those ligands, or by changing conditions like changing the pH or the salt concentration to disrupt the binding capability, and then releasing those uh, ionic complexes or those complexes in general. This can be used for purifying specific antigens. These can be used for purifying specific enzymes. These can be used for purifying specific hormones or signaling molecules, sorry, for purifying proteins with affinity tags.